everyone. So we'll get started. My name is Kelly Salmon. I'm the senior scientist at the New Hampshire Academy of Science. We're a nonprofit and we run a lab in Lyme, New Hampshire that is basically for middle and high school students to perform authentic scientific research and for training teachers to be able to bring that back to their own classrooms. Typically, we run a lot of after school programs and summer programs, and actually right now we are running summer programs remotely. So we've given equipment to students to take home, or they're doing computer-based projects, and we're advising them in their research as they do that at home. So that's a little bit about what we do, and then we also do a lot of outreach and things like this to really spread more information about science, particularly in New Hampshire and Vermont. So I'm going to be the main presenter, but my colleague Allison, who's also wearing this snazzy polo, and is Allison Michael in the chat. She's going to be acting as moderator. So if you have any questions, send them along to her and she will either get those answered in the chat or be the one that to interrupt and get us to go back a slide or whatever it is that needs to get done. So I think that is all the introduction-y things. Oh, and so this is the science of COVID-19. We're going to be doing this every Tuesday for all of July. So on the Claremont Makerspace website, who we're happy to be doing this collaboration with, it has all the topics for the rest of this month. And in particular today, we're going to be talking about PCR-based testing and how that's being used for COVID-19. And to give a summary, we're going to start with a little bit more about COVID-19 and SARS-CoV-2, the virus. Do a little bit of background on DNA and RNA that we're going to need to understand how these PCR-based tests work, and then really dive into how those tests are working and how they're being implemented in the clinic. And just as a caveat, my background is in cell biology, molecular biology, so I'm attacking this more from the science side than from the actual medical side, not a medical doctor. So the COVID-19, it stands for coronavirus, so co and vi, disease in 2019. And COVID-19 is then the disease, the symptoms that people exhibit, and it's caused by this novel coronavirus, SARS-CoV-2. And corona just means crown, so you can see from these images of the virus particles on the right that they have this crown or corona that's coming off from the virus itself. And that's actually the spike protein that we're going to talk a little bit more about today and even more in the coming weeks about, that's really sticking up off of the coronavirus, and it helps it attach to cells and get inside. Coronaviruses themselves are a large family of viruses, so there's quite a lot out there. They can cause the common cold, but as the name SARS-CoV-2 implies, they're also responsible for the SARS epidemic and also MERS, the MERS epidemic, Middle Eastern respiratory virus. And in humans now, we can see that COVID-19 disease symptoms range a lot from person to person, which is something that scientists are still trying to understand better, where some people are completely asymptomatic, meaning that they don't have any symptoms or anything obvious. They could have flu-like symptoms, either mild or severe, or they could have really severe respiratory symptoms and pneumonia. So it differs a lot from person to person. And just to go into what is a virus. So a virus is this little buddy over here. It contains a DNA or RNA genome, which we're going to talk more about in a little bit. And that's shown on the inside in this cutaway to the virus. And in the SARS-CoV-2 virus, it's single-stranded, which I've abbreviated as SS, RNA. And then that is the main genetic material, and that's enclosed in some sort of covering or envelope. And in the case of SARS-CoV-2, this is a lipid envelope or like a fatty membrane envelope. And that's why, in particular, washing your hands with soap and water is really good for eliminating the SARS-CoV-2 virus, because the same way you use dish soap to get grease out of your frying pan, it can help break up and remove the SARS-CoV-2 virus as well. And then they have a couple proteins that are on this outside bit that are just for helping the virus find where it wants to go, which in our case is into ourselves, unfortunately. and help mediate that interaction. And in in SARS-CoV-2, that spike protein, which is shown as this pink blobby dude here, binds to the ACE2 receptor on human cells, which is typically involved in regulating your blood pressure. And that allows the virus to get into the cell where it's able to replicate itself and go forth and go infect new cells. And this is one of the weird things about viruses, is that we don't classify them typically as being living because they can't reproduce on their own and they don't eat. So they just need to get into other cells, and they need to hijack that cellular machinery in order to make more of themselves and go out and infect more people or more animals, plants, or bacteria. There are viruses that affect every living thing. So because of that, we don't classify them as being considered as living. So I'm going to take you back possibly to like high school biology for a few minutes to go into the DNA and RNA that I've kind of mentioned with the viruses. So for DNA, that's what we consider as carrying all of our genetic information. And I've shown up here on the top a human karyotype. So in humans, all of our DNA is compacted down into 23 pairs of chromosomes. And this is what gets passed from mother or father to child that helps pass along traits. DNA is in all living things, which is why viruses are kind of weird. They can have RNA genomes. And again, you need that to make all the things that make you you. So all the DNA in your chromosomes is what makes your liver function, your lungs function, makes your hair color, eye color, everything about you. And on a structural level, if we really zoom in on these chromosomes down to the DNA, it is a double-stranded structure. So we have one strand, hopefully people can see my mouse, one strand on the left here with this backbone, we call it, of this little pentagon dude and a little circle dude, and then a letter in the middle. Oh, thank you, Allison, you can see my mouse. And then there's another strand on the other side. And this is kind of zoomed out a little bit more over here, showing you this DNA ladder. And these two strands are base paired in a complementary fashion. So there are 4 bases in human DNA: adenine, which is the A over here, thymine, T, guanine, G, and cytosine, or C. And DNA is such that the C always binds to G, A always binds to T. And this is really important because that means that for copying this DNA, we can separate those 2 strands and know from one strand what the other strand should look like. So that allows us to make an exact copy of the DNA just by splitting it in half and filling in the missing pieces. And then this whole thing folds up into this nice double helix that we see on the side. And I should say, DNA stands for deoxyribose nucleic acid. And deoxyribose just refers to this little pentagon dude, and it's the type of sugar that makes up the backbone. Then there's ribonucleic acid, or RNA, which compared to this double-stranded ladder that we just looked at, typically exists as a single strand, so it's not base paired. And it does still have cytosine, guanine, adenine, but instead of thymine, it has to be a little bit different, and it has uracil. So in RNA, adenine bonds to uracil. And RNA can still follow that same base pairing to base pair to other RNA or to base pair to DNA, which we'll not really talk about today. But this is also important for being able to replicate the viral genome as well, that it can make copies based on what it sees on the one strand. So how do we actually test for SARS-CoV-2? Well, in general, there are a couple of different ways that we can test for viruses. The main one that you often see in clinics is antigen detection. And antigen is just a fancy word for something that can be detected by an antibody, which we'll be talking more about next week. And that's part of your immune system that is able to recognize things that are foreign to your body. And that's what's being shown here. These, on this image, the yellow guys are antibodies. And they are trying to bind to the parts of the, in this case, coronavirus that they can see. And that's what we call an antigen. So the spike protein, which really sticks out from the viral membrane, makes a good antigen because it's easily seen by your immune system. And if you were to pick up a whole virus out of someone's respiratory tract, you'd be able to pick up that antigen using a simple test. This is often what you see used if you've had a quick strep test or flu test at a doctor's office. These are typically done with antigen detection. Then there's viral culture, which isn't really used as much. It's basically taking a sample from your respiratory tract or your blood, putting it onto cells and seeing if the cells show signs of being infected. So they start producing more virus. But this takes a while to do. It's more labor intensive and money intensive. So it's not as often used. We can test for antibodies in the person to see if they mounted an immune response to the virus. And we'll talk more about this next week. Next week is antibody testing and antibody convalescent therapy. So we'll be coming back to that one. And then what we're really going to focus on today, we can look for the DNA or RNA that is within the virus and use that as a readout for whether a person is infected. So when we do this, we first have to collect a sample from an individual, which this is the lovely swab that you've seen a lot on the news that basically goes back and touches your brain almost. And the reason why it goes back that far is to collect sample from your nasal passages, but also back in the back of your throat, which increases the chance that you'll pick up an infection that's really actively occurring mostly in your lungs. And for this test, it does only detect active infections. So again, antibody testing that we'll talk about next week is able to kind of turn back the clock and see if you have been infected in the past. And the important thing with this is that to actually be able to detect the virus, even if it's in low levels, we need to be able to amplify the genomic material that we're looking for. So to do that, we first have to get that RNA out of the virus. So when we do that fun swab, you get a virus or multiple viral particles if they're there. And you'll also pick up some of the actual human cells, so the cells in the back of your throat or in your nose that are part of the epithelial or like the lining of your nose and throat. And these will come back in a little bit later, but just think of them for now as a bystander on the swab that gets taken.
Kelly?
Yes.
Quick question. Why is it a nasal swab as opposed to a throat swab?
Ooh, that's a good, I have seen both being done. It seems like everyone's loving the nose swab that goes all the way back. But I have seen people doing both in like front of nasal, like just the inside of your nose, and a throat swab and running both as a test. I have to say, I'm not quite sure why it is the gold standard. And also for questions that can wait to the end, hold them to the end, but I'm going to write this down. So any questions I can't answer today, I'm going to write down and I will try to have an answer next week. Or at the end, I'm going to put up our email and you can email us if you would like to get a response sooner than that, or you can't wait, or you won't make it next week. So I'm writing that down. Okay. So once we have that swab with the virus, if it's there, and epithelial cells, we have to get the RNA out from the inside of them. And the way we do that, similar to the hand washing thing, is actually using a strong detergent. So that breaks up the membrane of the virus. It also breaks up the membrane of your more fragile cells. So not really the cells on your skin when you're using a regular detergent, but this is a very strong detergent. And that's able to break up the membrane of the cells and of the virus. And that basically punches a bunch of holes in it and allows everything that is on the inside of those cells and viruses to come out. And that's showing you here that we have DNA and also proteins and some other fun stuff from inside your cells. Then we only want the RNA from that. And this can be done a few different ways, either using these magnetic beads, which is kind of the most fun way of doing it, or using what's called a column, or basically a fancy filter. And in both these cases, you'll see silica is written here. In both these cases, it's using a silica filter, which basically using some fancy chemistry is able to bind DNA or RNA on the backbone that I showed earlier. And that's able to bind to that silica filter. And then we can wash everything else away and then reverse the chemistry that is binding the RNA to that silica. And that allows us to get back out at the end a tube of purified RNA in liquid. And an important note for this is you do get enough back out of this to run a test a few times. So this is going to come back in later. If something goes wrong in the test, they can rerun it. Then once we have that genomic material, we might not have a lot of it. So in order to make sure that we can actually tell that it's there, we can amplify it to get a whole bunch of copies. And that allows us to really definitively say, it was there and made all these copies, or it wasn't there because we tried to replicate it a bunch of times and it didn't show up at all. And I'll go into this in more detail in just a second, but the general overview is that you start with, so for the next few slides, I'm going to talk about DNA, and then we're going to come back to RNA. You start with your template DNA or your starting material. Then, similar to what I said earlier, where you can separate those two strands and copy based on that complementary base pairing, you can separate those two strands, make the copies, and this allows you to go in one cycle from 1 copy to 2 copies. Then those 2 copies can become 4 copies, 4 copies become 8 copies, and you get this exponential increase where after running the cycle 30-something times, 40 times, you can get billions of copies of your RNA. And what goes into this, this polymerase chain reaction, or PCR, is your starting DNA sample, primers, which I'll talk more about in a second, that marks the ends of what we're actually amplifying. So we're not amplifying the whole genome, we're just looking for a signature, a part of it. The A's, T's, C's, and G's, all those building blocks to make sure that we can make more copies. A Taq polymerase, which is just the protein that goes along that one strand of DNA, realizes it's sitting on a T that needs to be opposite an A, grabs an A out of the air, and binds that into the growing chain. And then it moves on, sees it sitting on a C, and it grabs a G. And this polymerase is called Taq. So polymerase is just a fancy enzyme name, meaning it makes a polymer. It takes multiple little bits, all these building blocks, and makes a polymer or a chain of them. And it's Taq because you'll see in a second, we're going to go through a whole bunch of temperature changes. And Taq refers to Thermus aquaticus, which is a heat-loving bacteria that lives in hot springs. And so that's been abbreviated Taq, and that allows us to use this with going through a lot of temperature changes. Then there's a buffer that just keeps the Taq happy. And all this goes into a tiny tube, which I actually have an example of. If you can see my video up in the corner, I am holding a very tiny tube close to the camera. So if I take that back near my glasses, you can see it's quite small. So all that goes into this tiny tube. We're working with pretty small volumes of liquid here, and goes into a thermal cycler, which just goes through those temperature changes that we're going to talk about. So there are 3 main steps to PCR. The first one is called melting or denaturing, and that's where we separate those 2 strands. So this goes up to 95 degrees Celsius, which is pretty close to boiling for water. And that melts these 2 strands apart so that we have 2 single strands. Then we're going to cool that back down quite a bit to about 55 to 70 degrees Celsius. And that's going to allow our primers to come in. So primers are what's marking the boundaries of what we're going to amplify. And the way that they do that is that there's small pieces of DNA that's single-stranded that is able to base pair to either end of what we're looking to copy. And it attaches to one attaches to one strand and one attaches to the other strand to ensure that we get copying across both strands. And you'll see that they're oriented going towards each other, so that way we end up amplifying what's in the middle of the two primers and don't amplify what's outside of them. Then we warm it back up a little bit to about 68 to 72 degrees Celsius, and that is the happy temperature for TAC. Again, being from a heat-loving bacteria, it loves being warm. So at that temperature, it's most active, and it's going to see those primers bound to the DNA and realize, ooh, there's double-stranded DNA that goes to single-stranded. I can fix that. I can fill that in. So it binds, and it starts pulling in the matching base pairs to go along that strand. And then at the end of that, we end up back at our template DNA, except now we're going to have 2 copies. And this will repeat over and over again about 30 to 40 times to ensure that we get lots and lots of copies of our DNA. But as I alluded to earlier, I've been talking about DNA for the past few slides, and we're starting with RNA from our virus. And the RNA is only single-stranded. So that presents a little bit of a problem, but not too much. So if we start with this single-stranded RNA, all we have to do is basically turn it into DNA. And we call this reverse transcription. So again, if you remember all the way back to biology at some point, you may remember that taking DNA and making RNA from it is called transcription. And here we're going from RNA to DNA, so it's reverse transcription. And scientists, being very creative, made the enzyme that does this, the protein that goes about and does this, named reverse transcriptase. And what it does is it sees a single-stranded RNA, and it's going to start pulling in the complementary DNA bases to make a double-stranded RNA-DNA hybrid that we call cDNA, or a copy of DNA. Then we can use this double-stranded now DNA template to do our PCR. And this process altogether is called RT-PCR, or reverse transcription followed by PCR. And when we do PCR in the lab, we're often treating it more as an endpoint assay, meaning that we're only looking at it after you've let the reaction run its full course. So in this case, we talked about that exponential increase in DNA that we get over the multiple cycles. So here on the y-axis is number of DNA copies across the number of cycles. We get this exponential increase, and then it tapers off again, basically because you've likely run out of your nucleotides or some other component, so it just can't do the reaction anymore. And it's normally at this point that we're actually looking at the PCR. And this is often done using a process called gel electrophoresis, where we are able to load that DNA into a gel, basically a jello-like structure that has little holes in the top. And then we apply a current across that gel. And I didn't mention before, the DNA backbone is actually negatively charged. So as you apply a current, it's going to move towards a positive end. And as it does that, smaller fragments move faster than larger fragments. So your smaller fragments end up towards the bottom, larger fragments towards the top. And we can use this as a readout for seeing if we got amplification in our PCR, and seeing what size the product is. Does it match what we expected? You can also use it to make some nice art over here on the side. And they love showing this actually on the news. They love showing people loading gels. So if you've seen these types of images on the news, that is from people doing gel electrophoresis. But in this case, we're not going to use that. And that's partially because if we wait till the end, number one, you have to wait till the end. which when we're talking pandemic, we want things to move quickly. And it also can lose small differences. So if we take that same type of graph with this exponential increase and a plateau, and we compare different reactions, there are going to be some that hit that really large increase faster, that had more starting material. So it was quicker for them to get to, say, this 1 million copies here. And then some are going to take a little bit longer to get there. So comparing this green line to this yellow line, if we look at them at the end, at cycle 40, they look the same. But if we look at them throughout the process, we can see differences. And so we can see this happening in action and quantify it using a type of PCR called quantitative PCR, or qPCR. And this can also sometimes be called real-time PCR because we're seeing it happen in real time. That then gets abbreviated as RT-PCR, which we just talked about, different RT-PCR. So that gets a little bit confusing. I'm going to stick to calling it qPCR. And just as fun science nomenclature, you can also sometimes see it as qRT-PCR or RT-RT-PCR. But this qPCR allows us to monitor each tube, so each of these little guys as they're going through the reaction, and actually watch that increase in DNA copy number in real time. So we can capture what's going on in these earlier cycles and compare from tube to tube. And how do we actually do that? How do we see that? We use a fluorescent dye. So in each of these tubes, we've also added, in addition to those PCR components I showed earlier, a type of dye called, typically the brand name is Cyber. And that binds in particular to double-stranded DNA. And importantly, it only fluoresces when it's bound. So when it's just bopping around in the tube on its own, it's not fluorescing. But as it binds to double-stranded DNA, it's going to fluoresce. And as we go through the PCR, we get more and more copies of that double-stranded DNA. And then more and more of the Cyber is able to bind and fluoresce. And so typically we use this sort of non-specific dye. We can add another layer of specificity by linking that fluorescent molecule to a probe or a sequence that's specific for the viral gene that we're amplifying. So this qPCR can give us a relative concentration of our starting material, the starting RNA, based on when the level of fluorescence crosses a threshold at which we can say we definitely have fluorescence, we have amplification. So this graph has now changed a little bit to show relative fluorescence on the y-axis, and number of cycles is still on the x. And we've added this yellow bar for the threshold. So below that, we might not quite know that it's fluorescing for real, but once it crosses that threshold of fluorescence, then we can say that it is definitely amplifying. And the point at which our amplification line crosses that threshold is called CT, for crossing threshold. So then if we take this graph with more lines over here, we can see that the CT number for this green line, where it crosses at about 16 cycles, that is much lower than this yellow line at the end, which doesn't cross the CT line until about 30 cycles. So why are people using qPCR for this? First, it can give a yes answer faster than waiting till the end and running a gel, because you're adding that extra step in running a gel, and you have to let the reaction go to completion. And it can also, if we take this quantification, it can give a relative amount of viral load, which, as we're learning more about the virus, this can be informative to compare patients based on how much viral load or the amount of virus that they have that we know of. And the original tests for were for PCR also because that's the fastest thing to design a test for. So all we need that is specific to the coronavirus is those primers. And once you know the sequence of the coronavirus, which was solved really quite quickly, in the pandemic, or before it was even labeled a pandemic. Once we know that sequence, we can design primers, have those delivered in about 2 days, probably under 2 days if you're close to a source, and really get going with your testing. And like in all science, when we do these types of tests, and you want this in all your medical tests too, you need controls. So alongside each of the patient samples that is being run, or every batch of patient samples that's being run, is a positive control that we know contains the RNA for SARS-CoV-2, and a negative control, a tube that we know doesn't contain that RNA. And this is to make sure that our assay is working like we think it should. So the positive control should show up as being positive. The negative control should test negative. And if either of those shows up wrong, if you get a signal for your negative control or you don't see anything for your positive control, you throw out your whole test for all those patient samples. You go back to those starting tubes of RNA that we purified and you run it again. Because you don't want to trust it if you think maybe your positive control didn't show up. Maybe you forgot to add your Taq polymerase and all your reactions failed. So you can't call those patients negative. You have to go back and rerun the test. Something that is also being done a lot in the testing for this is in the same qPCR, you can actually use a different color fluorescence if you're using those sequence-specific probes. And you can test for a human gene at the same time. So if we go back to when our swab picked up both virus and some human epithelial cells from inside your nose and throat, if we want to make sure that basically it's guaranteed that if you have a swab up your nose, it probably picked up some of your cells. So we want to make sure that we were able to successfully extract and amplify the RNA from those cells. And if we don't get any signal from either a virus or the human cells in that case, it might be that that swab wasn't used correctly, or it was not stored correctly in transferring it to the lab and everything degraded possibly. So these swabs have to be transferred cold to maintain the integrity of the virus and the RNA. And if, say, it was out in a heat wave for a while, the sample may have been destroyed and you're going to need to go back to that patient. So when we think about this qPCR test, there's some important things in terms of time and needs. So this is not a very short test. It's not like the rapid strep test. You have to swab your patient, get that swab transferred to a lab, set up the RNA extraction, which takes about an hour, set up your RT and your qPCR, also takes about an hour, and actually run the RT and qPCR, which is about an hour and a half. And all of this needs a nice lab that's well equipped, trained scientists and technicians to run all these samples, all the reagents involved in the RNA extraction, RT and qPCR, and importantly, it takes a lot of time. So this is where, using the same general idea of amplifying genomic material, we get the Abbott rapid test. And you'll hear this called both a 5-minute test and a 15-minute test, or a 13-minute test. And that's because it can give a positive result in as little as 5 minutes. If we think back to seeing those curves increase, if you have a large viral load and you get that increase pretty quickly, it can be as little as 5 minutes, or it will run out to 13 to 15 minutes if it's going to be negative, but make sure to run its full cycle. And the reason why this is faster, or part of the reason why this is faster, is it uses a different type of DNA amplification called isothermal. So polymerase chain reaction, we cycled through those temperatures. Isothermal DNA amplification all occurs at the same temperature, which means we don't have to cycle through. And as you start getting more and more copies, everything can be happening simultaneously, rather than everyone needing to be in the melting stage, then everyone in the annealing stage, and everyone in the elongation or extension phase. This system is also really easy to use. So it has these nice little pre-made cartridges, which basically in the purple part of this machine, you're able to do your RNA extraction. Your RNA extraction by just swirling your swab around, goes into this nice buffer that lyses all your cells open. And then you're able to move that into the orange cartridge that goes up in the orange area. and that's where your DNA amplification is going to occur. And so this doesn't require the separated RNA extraction followed by RT-PCR situation. And in this case, the detection is also fluorescence-based. So this machine is also reading for fluorescence over time as it runs the reaction. And that's using a sequence-specific probe, so something that specifically binds to the RNA sequence and fluoresces. And as you get more and more copies of that sequence, the level of fluorescence goes up. So in summary, I've talked to you today about these RNA tests using PCR, and these can detect active infections only. They can't tell if you've been infected in the past, but this can actually detect early infection. So on this graph on the right, the blue line is showing you the RNA and antigen, basically the amount of virus you have in you, and that goes up as your infection goes on. And in this early period, using the PCR-based tests, we can actually get a positive result on a person who is infected. And then as you get more and more virus, your immune system, which we'll talk more about next week, starts kicking in, and then you start getting antibodies, which are classified out into IgM and IgG. So these antibody tests detect later infections. They'll miss this early time before the immune system starts ramping in. But they can show when you have a later stage infection and show that you have been infected in the past. And again, we'll talk a lot more about that next week. And with that, I would like to thank the Claremont Makerspace for allowing us to do this collaboration and giving us a venue. I'm with the New Hampshire Academy of Science. Our website is here, and also our primary email. So if you have any questions after this, or you want to follow up on something or contact us about anything else, you can find us there. And we're also partnered with our satellite laboratory at the Fairbanks Museum and Planetarium STEM Lab. And we can open it up if people want to ask questions.
Yeah, we have one question already from Leanne. She'd like to hear about the different strains and how testing is affected, and can they test for multiple strains at once?
So to my knowledge, they're testing for kind of a broad enough, or a part that will not change as much from strain to strain. And I started looking into this, and I wasn't able to completely follow through on my literature reading desires. And I will write this down too. So now there is a lot more conversation about different strains, because we're starting to wonder if maybe we have switched to a different strain that might be more infectious, maybe less deadly. So that is coming up more and more. So the way that we normally design primers for doing PCR is, unless you want to specifically see changes between strains, you will stick with areas that you know are going to be pretty consistent. So I'm going to make a human analogy to this virus situation, where if you're just trying to see if you were infected with a human, you could look for parts of the genome that vary a lot from person to person, or you could look for parts of the genome that are really similar from person to person. So an example in humans would be our hemoglobin gene that carries oxygen in your blood. That is really, really consistent in its sequence between humans. And that's partially because it's really important for you to live as a human. You need oxygen in your blood. So because that gene is so important for us, it tends to not change a lot from person to person. And so if you were looking for an infection of generic humans without caring about what type of human you were infected with, if you're infected with Bob or Sue, then you would look for something like the hemoglobin gene. So from my understanding, for the viral tests, they're looking for basically that kind of equivalent, something that won't change as much from strain to strain, because it's really important for the virus. And they won't focus as much on parts that can change a lot. But for actually detecting strains and getting more information, a lot of times these patient samples are then sent for full sequencing. So you know the person has the virus, you have everything extracted, and then you can go and sequence the whole genome. And scientists are depositing these in databases online so that way people can compare strains that are popping up in different areas and try to correlate that with patient outcomes and human health. Anybody else?
I don't have any more chats.
Okay. So people can chat at the everyone or chat at Allison or chat at me now. Or you can unmute yourself and chime in if you would like. Okay. So, oh, thank you. So as I said, next week we're going to be, and as I teased a few times throughout this presentation, next week we're going to talk about, very broadly speaking, the immune response to viruses, and then in particular how that relates to antibodies in terms of antibody testing, and also what it means when they talk about convalescent antibody therapy as a type of treatment. And then, if I'm remembering correctly, I live day by day. The following week, I think, is vaccines. And then at the end of the month, Allison's going to come in with her chemistry background and talk about what role protein modeling and actually looking at the structures of these different proteins is playing. And how you can do some of that yourself, either by fiddling around online using different servers or donating your computer to have someone else run their experiments.
We do have a question from Jessica Warren. She wants to know what leads to false positives or false negatives.
Ooh, that's a good question. So false positives, I actually talked to someone who does this type of testing. It can be a lot of just accidental contamination around the lab where they're running these tests. So say in a hospital, you have a testing center that is doing a whole bunch of these tests at one time. And though they're being really careful, it's possible that you could get some cross-contamination when you're setting up your samples, or sort of mysterious airborne magic things that might fall into your tube. And that can give you a false positive, in addition to things like your samples failing for some reason. And then false negative, I've seen it stated a lot for testing really early on, before a person really has enough, like maybe the swab just didn't get to the right part. And especially the swab that goes way back and touches your brain is probably better at, and I should clarify, it doesn't actually touch your brain, is better at picking up things that might be going on in your lungs than some of these tests do swabs that just go in the front of your nose. So from what I've seen, a lot of the false negatives tend to be earlier testing, that it just doesn't have enough material to work with yet. Okay. Thank you everyone for coming. We really appreciate it. We hope we will see you again later this month. Thank you guys. Have a good night.